Hello guys, today we're going to talk about the lymphoid system. So we face two different structures, the primary structures and the secondary structures of the lymphoid system. The primary is the thymus and the bone marrow, and the secondary we'll find the tonsils, lymph nodes, spleen, and MALT. MALT stands for mucosa associated lymphoid tissue. So just a quick intro to this. The red bone marrow is the source, is the location, the organ which is responsible for the production of all the precursor cells of the immune system. So even a T cell, which is actually matures and differentiates in the thymus, the precursor cell of the T cell is produced in the bone marrow. And the thymus is the organ responsible for the maturation and differentiation of this precursor cell into a mature and functional T lymphocyte or lymphoblast. So the bone marrow, of course, is the location where the B lymphocytes and lymphoblasts mature and grow. So let's start getting into some basic introductory elements of the lymphoid system, and then we can continue on to the specific organs. Of course, this is going to be a quick intro for detailed information about this adaptive system, innate system, and generally the whole immunological structures in the lymphoid system. You should, of course, check out books of immunology, textbook immunology. As an example, one very good book is the Basic Immunology of Abbas. Incredible book, and contains many interesting things that you need to know as a medical professional. So let's discuss about the innate and adaptive. What is the difference? The innate system is the system that is responsible for the countering and for the that is against non-specific pathogens. It doesn't matter what type of pathogen it is. It could be virus, it could be bacteria, it could be any sort of pathogen. The reaction is going to be exactly the same. For example, the skin is a human barrier. This is one part of the innate system that provides protection against any sort of mechanism. Also, the enzymes that the skin produces are antimicrobials. This is, for example, one very short example of the innate system. And adaptive system is the specialized, very specialized, highly accurate system of countering bacteria, pathogens, viruses, and so on. For example, the contents of this adaptive system is the T lymphocytes and the B lymphocytes, as well as, of course, all the products and all the molecules related to this function. So we have, of course, cellular and non-cellular elements in both cases. For example, in the non-cellular element typical for the innate system is the complement system. Again, the system should be highly studied and you should highly focus on the system and learn a lot about it through, of course, immunology textbooks. So we're going to talk about some basic terms such as chemotaxis. Chemotaxis is the procedure during which any sort of white blood cell is gonna be drawn into a specific site of inflammation or any sort of signaling process. This thing is that, in simple terms, it means that chemotaxis is the procedure by which any chemical molecule can induce a movement of a white blood cell. Now, the chemotactics are molecules that are a subcategory of cytokines. We've already discussed cytokines numerous times. Nonetheless, cytokines are molecules that induce movement of cells from one location to the other. And of course, one subcategory of this of the cytokines is the chemotactants. Now, we have the antigen. What is the antigen? Antigen is, by definition, anything that can be considered foreign to the body that it actually invades. For example, any normal human structure. Let's take, for example, the skin cell, okay, and a keratocyte. If you take keratocyte and you place it in a different organism, for example, a goat or a rabbit or whatever other it is, this is going to be considered as an antigen. Now, typically, of course, antigens are mostly proteins, are mostly glycoproteins, saccharides, and many other molecules. But any molecule that is foreign to this specific organism that receives this substance is considered to be an antigen. Now let's talk a bit about the antibodies and the two different portions. We have, of course, many different types of antibodies. We have the IgMs, IgGs, we'll discuss them in a bit. The point of this discussion is that there are two different locations of the antibody. There are two different loci, let's say. One is the Fc, the constant portion. For example, let's take the IgG. Now, these are the different categories. We'll discuss them in just a second, but just focus on the typical picture that we have of the antibody, which is exactly this one. So the constant portion is this bottom part right here. This is constant, and it's called constant because it's exactly the same in all the different antibodies. Just keep in mind that antibody is a specific molecule that induces and neutralizes parts of the antigens. So then this happens by binding, and we'll see also with many other ways that it can happen. But the point is that it has to be highly specific. So we have these two portions, the Fc, the constant portion, which is exactly the same and never changes, and we have the F antigen binding section, FAB, exactly on this point. The top part of the antibody is this highly specific towards an antigen. So we have exactly these two areas of the antibody that you should absolutely know before we move on. Another feature you will see is the MHC. This is the major histocompatibility complex. This is nothing more than just a group of receptors that are being shown into nucleated cells. Now there are two different types of complexes, the MHC1 and the MHC2. The MHC1 is just nothing more than just specific receptors in the cell membrane of all nucleated cells, and the point is that they actually present self-antigens. Meaning what? For example, let's take a normal healthy human cell. This healthy cell is going to actually disintegrate and destroy different components of its inner contents, and of course is going to provide this part of a protein or whatever it is, part of protein, to this receptor, the MHC1 receptor, and expose it in its cell membrane. This is useful. Why? Because we can understand its usefulness when it comes from a pathology. For example, we have a virally infected cell. So as a consequence, it's very common that this disintegrated content of the side of the cell, for example in this case will be a viral antigen, is going to be presented into this receptor again in the plasma membrane. And as a consequence, the adaptive system, we'll see how, will eventually induce a response, an inflammatory response, and of course to counter this exact specific viral infected cell. And also we have the MHC2 complexes. Now what are these? These are special complexes, these are special receptors, again similar to the first, but they're only present in very specific cells. The cells that we're talking about in this case are the antigen presenting cells, or also called APCs. Now there are two categories of APCs. There is this professional and the non-professional. For example, we have a typical professional APC is the macrophage. The macrophage, by its nature, phagocytoses some pathogen, and in the end, after cleaving specific portions of this enzyme, will actually present this antigenic parts of this pathogen that it phagocytosed, and expose them and express them in its cell membrane. And this, of course, will, as a consequence, lead up into the specific inflammatory response and actually cause, as a final result, the production of antibodies and the production of humoral and cytological response from the adaptive system. We have also non-professional antigen-presenting cells. For example, the fibroblasts. The fibroblasts can also play a role in this procedure. Of course, they can phagocytose something and eventually express these antigens again in the cell membrane, which as a consequence will induce a whole immune reaction. So let's see actually what is the function of the antibody, just a bit. Before we do so, let's see the different types of lymphocytes that we have in the human body. We have the T lymphocytes and the B lymphocytes. Now there are specific subcategories we should discuss in the T lymphocytes. We have the T helper lymphocytes, CD4 positive. CD stands for cluster or differentiation, and again this is simply specific receptors in the cell membrane of the T lymphocyte, the specific one. We have the cytotoxic T cells, T lymphocytes, and in this case these are CD8 positive. This is again cluster differentiation number 8. And also we have the regulatory T cells. These are CD4 and CD25 present positive cells. Now what are the functions of these cells? We'll have the T lymphocytes, the helper T lymphocytes, the CD4, which actually play a major role when it comes to the production of antibodies. They play a role into the communicating role between the connecting point between the T lymphocyte and the B lymphocyte. Meaning what? Typically T lymphocytes are responsible for the cytotoxic, of course, reaction to the cell. For example, we have a virally infected cell. The T helper cell is going to induce, of course, a reaction, and in the end, the CD8 positive, the cytotoxic cell, is going to induce apoptosis to the cell, hence neutralizing the virally infected cell. So the T lymphocyte is, of course, the connecting point between the T lymphocytes and the B lymphocyte. Typically, let's say that cell is infected with, let's say, some other sort of, like we have a bacterial infection, and we have the antigen presenting cell, the macrophage phagocytoses this part of bacteria, and this bacterium, this part of this antigenic component of the bacterium is going to be presented in the cell membrane, and as a consequence we're going to have the activation of the CD4 positive cell. Now the CD4 positive cell is going to, of course, grab onto this macrophage, this activated macrophage, and induce and produce cytokines, which will eventually activate the B lymphocyte upon this activation of this B lymphocyte, we're going to have the differentiation to plasma cells and then memory B lymphocytes. Meaning what? Of course, the plasma cells are the cells that are responsible for the antibody production. We already discussed this in the connective tissue videos. But the point is that after this activation of the B lymphocyte, we're going to have the differentiation to plasma cells and of course, the memory cells. The memory cells, the memory B lymphocytes are going to be cells that will reactivate upon a second presentation of the same antigen, hence leading to a very much faster response from the human body. So the regulatory, the CD4 positive and CD25 positive cells are the cells that are responsible for modulating and terminating a specific inflammatory response. Without these cells, an inflammatory event would actually take much longer, leading to devastating effects. You'll see later on in pathophysiology that most of the pathomechanism most of the bacteria, most of the pathogens that induce damage to the human body are actually also indirect cause of the excessive activation of the inflammatory markings. You'll see many times that excessive inflammatory findings that actually are the ones that cause the pathology to the specific disease. So there has to be a very nicely and very tightly regulated inflammatory response. Up to some point, it can be very helpful to the human body to combat some specific pathogen, but in other cases, in some specific cases, of course, we need to immunosuppress this reaction. You'll see this one further on in pathology and so on. So these are, of course, the different categories that we should firstly know about the adaptive cells. Now, let's talk a bit about the different forms of the antibodies. There are many different forms. Specifically, there are 5 different forms of antibodies in the human body. Of course, each specific form is unique in its own different way. Let's see what it is and how they're actually different. First of all, we have the IgGs. The IgGs are the most common antibodies in the human body. And of course, in this case, the more numerous, of course, and these are actually very useful for many reasons. First off, these are the smallest antibodies present in the human body. That allows the size, of course, the small size of these antibodies allow these antibodies to penetrate many different barriers. For example, the fetal circulation. We have the barriers between the mother and the embryo in this case, and of course this is a barrier that is very specific and very tightly controlled and regulated. This means what? This IgG is the only possible antibody that can penetrate this barrier and allow for the human embryo to combat a specific pathogen or some specific let's say infection, whatever it is. And this is the only one that's actually available and able to pass throughout this fetal circulation. The functions, of course, of all of the antibodies is, of course, it activates phagocytosis, neutralizes antigens. We're going to see this shortly in a bit, just the very nice table again from Juncker that actually shows all of these reactions and all of these activate the results of the activation of the antibodies. So, also further, very important to notice is that IgG is the second antibody produced after an infection. The first antibody produced against an acute infection is the IgM. The IgM again is the first one that's being produced, meaning what? Meaning that whenever we have a specific antibody testing, a specific like checkup test, and we can see the proportions of the IgM to IgG for a specific, let's say, antigen, for a specific, let's say, hepatitis virus, we can actually time and understand the timing of this infection. If we see a high amount of IgM, this means that this is an acute infection, for example, in hepatitis. If we see a high percentage of IgG, then this means that this is a more chronic or at least longer lasting infection and infection of the specific pathogen. So the IgM, this is a very important molecule because it's a very big molecule. In fact, it's a pentamer, meaning that it has specific 5 monomeric points of the antibody. In other words, we have 5 FAB portions, 5 portions of the antibodies that actually bind the antigen binding sites. So this is actually the first, as I said before, and activate the major one of the major functions of this antibody is that it activates the complement system. Then we're going to see the IgA. This is the mucosal antibody. Of course, we have many mucosal surfaces in the human body, the mouth, the skin, and so on, the GIT tracts, anything that is actually in touch with the external environment. Let's take, for example, the human mouth. In the human mouth, of course, there are many secretions. We can find this IgA in many secretions, saliva, milk, tears, and so on. Again, all of them are present in the mucosal surfaces. And of course, the point of this is a dimer. We have 2 different FAB portions in this case, and we can find this antibody present in the mouth, and again, as we said, in the mucosal surfaces. Unfortunately, the percentage is much lower than the IgG, but still it's efficient in a very many numerous cases. So we have the last 2 of these, the 2 different, one of the 2 smallest proportions and 2 smallest numbers in the human body, in the plasma specifically, the IgD and the IgE. Now, we've already discussed the IgE, and we said that the IgE is present typically in the surface of the mast cells and basophils, and in this case, this is mostly related to their reactions against parasites, worms, and most importantly, it participates in allergic reactions. Lastly, we have the IgD. This is the rarest form of the antibody, and this is simply present in the surface of the B lymphocytes. So let's see actually what is the consequence of what are the activities and the results after the activation of the specific antibody. First of all, we have the neutralization. This is very important because in the case, for example, of a viral molecule, as in the picture seen exactly in this picture, one of the most pathogenic causes of the virus is the ability to bind on a specific human cell and invade it, and hence multiply and produce more viral molecules and more viral, let's say, factors. In this case, when we have a specific antibody, this FAB portion is going to bind on these specific receptors and specific pathogenic points in the cellular membrane of the virus, hence neutralizing it, hence stopping and inhibiting the process of viral attachment from the virus to the specific human cell. This is one result that we can have. The second is the agglutination, the clumping of the bacteria. Now, this is the second point. This is a cross-linking procedure between many different antigens and clump as a consequence, forming this mass of bacteria specifically located in another area. Now, we have also some other toxins. For example, the toxins are molecules that are being produced by bacteria, and just these small molecules are enough and are able to cause true pathogenic consequences to the human body. As a consequence, one of the very few, let's say, barriers to stop this toxin result of toxic functions is of course the antibody. The antibody can bind specifically to any sort of molecule and practically stop it from inducing a specific pathogenic reaction. Now let's go to see exactly the reactions of what happens when we have an exposed Fc portion. What happens when the antibody, the binding site, is already activated, it's attached on this specific antibody, and what can happen exactly after that? First of all is the complement activation. The antibody, the Fc portion, is able to actually induce the complement activation, and as a consequence cause the lysis of a bacterium or any sort of other pathogen. We have also the opsonization. What is the opsonization? Opsonization is opsonization is the process of tagging any sort of pathogen, and in the end we're going to have the phagocytosis of this tagged molecule, tagged pathogen. For example, we see in this picture right now, we can find the phagocyte, the macrophage specifically, that already has, let's say, has grabbed on these to the antibodies. In this case, the antibody, of course, is attached already to the bacterium. So because of the Fc portion is exposed, because of the presence of the exposed Fc portions in this antibody, we have the activation of the phagocyte and induces a faster and more efficient phagocytosis of this specific pathogen. And lastly, we have the activation of NK cells. NK cells are called natural killer cells. These are typically cells that we can find. This is part of the innate immune system, and we can actually use NK cells attack and specifically destroy virally infected cells. We can also find the NK T cells, that are a special subcategory of T lymphocytes. Another special subcategory of T lymphocytes that we should mention are the gamma delta T lymphocytes, also called intraepithelial cells, intraepithelial T lymphocytes, because of course, as the name suggests, they're present again in the mucosal surfaces, specifically more commonly found in the human skin. So we have one common feature throughout the whole lymphoid system, the presence of reticular elements. And I'm being specific and I'm saying reticular elements and not only reticular fibers, because we can find some exceptions to this rule. So nonetheless, in all the cases, in thymus, in the lymph node, in the spleen, we will always find reticular elements. So let's take, for example, the lymph node. The lymph node is filled with reticular fibers and reticular cells. Just again to revise, to bring it up again, to say just once more, reticular fibers is nothing more than simply collagen type 3, and the reticular cells are nothing more than specialized fibroblasts. And in these cases, both the reticular fibers and reticular cells are argyrophilic. They are silver. They can be silver. They can be stained, especially with silver staining impregnation methods. Let's actually visualize the proportion of how much of these elements are visible in the lymph node. For example, this is a very nice section of the lymph node, and we can see this bottom picture. This is only the silver staining method, and of course, we have the positivity in the presence of the reticular fibers and reticular cells. We can actually visualize the intensity and the amount of such reticular elements throughout the whole slide. What is the point of this? Of course, reticular cells and reticular elements are, as the name suggests, are networking elements. They're molecules that allow and induce, let's say, a better communication between the antigen presenting cells and the lymphocytes present. Why is it so necessary? Well, because all of these reactions of the adaptive immune system are caused as a consequence of close binding from the antigen presenting cell to the lymphocytes. If we don't have efficient binding from the macrophages or any sort of antigen presenting cell to the lymphocyte, then we cannot actually arm and we can actually have a very efficient adaptive response to it.